Hello, everybody. My name is Roman Bauer, and I'm a computational neuroscientist at the University of Surrey. Today, I'm going to talk about some of our research where we use simulations of brain development to explain neural connectivity. So the brain is an extremely complex organ. Here you see a visualization in 3 dimensions where different colors indicate different orientations of fiber tracts in the brain, as estimated or created with diffusion tensor imaging, which is a type of MRI method. So this connectivity is visibly very, very complex. And if you want to understand how to simulate the growth of such complexity, we need to also understand the underlying fundamental principles, of course. And these principles are based on the interaction between 2 entities. One is the kind of gene expression or cellular behaviors that interact with the local external environment of a cell body, for instance. So a cell can, for example, grow and migrate and so on. And it does this by utilizing information that is available at its local cell body. And it can also change the information and write on this local environment. And so essentially we have cellular behaviors that interact with the information that is available in the local environment. If you want to simulate such models, then we can follow the essential basic approach of computational modeling, where we have a certain system, a target system that we are aiming to model. And we can then create a model and configure that model. And of course, we need to validate it. And we do this by comparing with experimental data. And then what is also very important in this type of approach is that we can create certain predictions that were not included in the validation. And so those predictions then need to be, again, confirmed with new experimental data, right? And so if these predictions are not are found not to be accurate, then we need to go back to the drawing board essentially and change the model or refine the model and really understand what may have been the issue. However, if the predictions are confirmed, then we can go back to the model and maybe increase the detail or increase the model size and extend the model in order to address more complex questions and more important questions. So we can essentially iteratively do this, and so we can more and more increase our understanding and improve essentially our models, our computation models. So this is the overall approach, and I want to now give you an example where we did this approach, and to this end we looked into so-called hubs. So what are hubs? Hubs are nodes in a network that have a lot of connections. So these nodes participate in many links, and the number of those links is much higher than you would expect based on a random regular network. So in the brain, it has been confirmed in the human brain as well as many, many other species. It has been confirmed that if you look at the brain regions in the brain, then indeed some of those areas are actually hubs. They are really highly connected and much more so than you would expect from normal networks, regular networks. And also, in addition to this anatomical interesting aspect, is the fact that those hubs seem to be implicated in many disorders of the brain. And we will actually get to that also later on. But for example, they are implicated in Alzheimer's disease as well as other types of serious conditions. And if you look into this more mathematically, then we can essentially plot on the x-axis the degree of nodes. So the degree is how many connections a node participates in. And on the y-axis, we have the chance to see such a node. If we have a random regular network, then we would expect something like here in red, where we have a type, almost like a bell curve, right? However, in reality, in almost all neural connectivities that we observe, we can see a different shape, particularly where the tails are wider, and so if you look at the higher end of the degree, this means that there are more hubs than you would expect. I mean, you may observe some hubs also in a regular network, but it's very much less likely than you would do in a for example, in a human brain of interareal connections. So this is just to kind of visualize this also from a more mathematical perspective. Now, how did we address this question of hubs? How those hubs develop? Well, we came up with a model called the nonlinear growth model, and this nonlinear growth model is inspired by bacterial growth as well as, for example, Ebbeson's parcellation theory, which is a theory about how the number of brain areas increased during evolution of the brain. And in contrast to other models, to many other models, here the number of nodes that are added to the network during growth is not linearly increasing. So it's not like we add a new node at each time step, so 1, 2, 3, 4, but it's actually non-linearly increasing. And that's why it's called the nonlinear growth model, because you have an exponential curve here. And so what you then would expect is that those nodes who were there very early on, they are much more likely to become hubs because they've more time than the others, than most of the others, to connect and form connections with other nodes. And so that's the underlying principle of the nonlinear growth model. We then assessed this model based on 4 real-world data sets, in particular protein interaction networks. We also looked at interareal brain connectivity in the macaque monkey. We looked at synaptic connectivity in the C. elegans, and we also looked at the non-biological network, which is the international airlines connections between airports. So we looked at the network of airports that are connected via operating airlines. And so then we compared our model, the nonlinear growth model, with 2 other models, which are very well established. One is the so-called preferential attachment model, also sometimes called the Barabasi-Albert model. And there you can also create hubs, for example. And then we also looked into comparing with the duplication divergence model, which is inspired from the evolution of proteins and genes. And so what we also would like to highlight is that the nonlinear growth model is biologically plausible because it does not assume any non-local information exchange. So in contrast, for example, to the Barabasi or to the preferential attachment model, there in the model, there's a term that requires knowledge of all the connections in the system. And so this is problematic, of course, from a biological perspective, because if a new connection is formed and that connection needs somehow access to some knowledge of the entire system that is not available necessarily locally, then that is an issue, that is a problem, right? And so our model is very simple and does not require such non-local information exchange. So what we then did is we looked at different features of those real-world networks, like, for example, the coefficient of variation of the degree distribution, which measures how wide the degree distribution is. And then we also created our own measure called the Hub-Rich Club coefficient, which measures tendency for hubs to preferentially connect with one another. And we then optimized those three models in order to get as close as possible to the real world features. And we had the same number of parameters for each model. Notably, we also created two different versions of the nonlinear growth model. I will not go into much detail here, but essentially we had the same principle of nonlinear growth, but slightly different ways of creating connections. And so that's why we have an NLA and an NLP model here. Now, importantly, we also looked into the maturation trajectory of the hubs. So if one can, of course, look at the final network only, right? One can look at the certain types of features of the final network connectivity. But what we did here as well is that we looked into when during the maturation of the network or the growth during the network, certain nodes appeared. So for example, in the C elegans, we know when which neurons develop. And so what we did is we categorized them into three different time epochs. You can call this time or time bins. And the same, for example, for airline between airports, we know when the airports were constructed. And so we can essentially say, okay, some airports are much older than the others. And so we can then also look into when the hubs actually develop. And then we optimized the models to get as close as possible to the observed maturation trajectory. Now, this is a summary table of these optimization results. So vertical bar indicates that a model can reproduce coefficient of variation. Horizontal bar indicates that a model can reproduce the Hub-Rich Club coefficient. And the square indicates that the maturational trajectory can be well reproduced. And so you can see here that there's not a single winner that can do everything better than the others, but so it's more complex than that. But you can clearly see that the NLA model can reproduce always those two features, and the NLP model can always reproduce the CV value and the maturation trajectory. So depending on which model you choose, you can optimize for certain types of features. And so this is essentially a way to demonstrate that our nonlinear growth principles can serve also as a benchmark model to compare with other models in the future. Right. Then, as I mentioned before, those hubs are also implicated in certain disorders, and the hubs, they form a certain organization called the Rich Club organization. So this is an organization that denotes the fact that hubs in brain networks tend to prefer to connect to one another rather than to non-hubs. So that's why it's called the Rich Club organization, because sometimes wealthy people like to connect preferentially with other wealthy people. And here you see that also in the brain, those nodes that have a lot of connections, they like to connect to other nodes with a lot of connections, much more so than based on chance. And what is interesting is that it has been observed that in the preterm born brain, that the hub, the Rich Club organization is even more pronounced than in the control brain. So this is a little bit counterintuitive. You would usually expect maybe that it's less pronounced, but actually it's the opposite. And so this is an interesting question, right? It's like, can you create a model that explains why this may be the case? And so what we did is we extended our nonlinear growth model to incorporate also weights. So we have also strengths now. It's not only binary connections, but it's also strengths, because we have that information in the human brain from diffusion tensor imaging. And then we could, by using this model, also generate essentially preterm brain networks, so simulated preterm brain networks. These are all simulated samples, which have a more pronounced Rich Club index. So this is just to show that this kind of simulation approach can also help with better understanding certain biomedical questions. And at the moment, we actually work on more detailed, more sophisticated modeling, and we are including also more real-world data to this end. So these are all these were some examples. Here there is a publication that we recently had where we present adaptive rewiring, which is a modeling approach or principle for neural network development. So people who are interested in this kind of approaches are encouraged to have a look at this recent paper where we give an overview of this adaptive rewiring, which is very relevant in this context. Now, these were all rather abstract models. Of course, it may be that we want to create more biologically realistic connections, and so we need to then also look into how is a neuron built, right? So in a neuron, we have a cell body, we have dendrites, we have the axons, we have axon terminals that then connect to other neurons. And if you look at networks between individual neurons, we get very complex organizations also here. And this then requires some dedicated tools, computational tools to simulate such growth. And this is exactly what Biodynamo is doing. So Biodynamo is an international consortium. I'm the spokesperson of this consortium, and we are currently 8 members. And we have created a software that allows to simulate highly detailed and sophisticated neuronal growth models, where we can really simulate biologically realistic morphologies and connectivity on a high level of detail. And also this Biodynamo software is very performing from a computational perspective. And just to give a kind of an illustration of this, so here you can see many thousands of neurons that grow and that really generate this kind of morphologies that are much more closer to the actual neuronal anatomy that we observe. And we also published in the past several relevant papers with Biodynamo. For example, here we published a paper where we focus on a particular type of axon, so-called thalamocortical axons. So these are long-range axons that then branch in this case in primary visual cortex. And so we looked at real reconstructions from such axons, and we created optimized growth models for this kind of model, and then we can visually and also statistically, quantitatively compare, and we could really generate very realistic neuronal morphologies. And of course, if you get the morphology right, then you are also getting closer to get the connectivity right. And then ultimately, we are aiming for some also functional simulations, and we have currently several lines of research along in this direction. Another recent paper that we published is here. That was just last year, where we used approximate Bayesian computation to optimize various features of those neuron growth models. So when you grow a neuron, you have many parameters, of course, and we can using approximate Bayesian computation, we can actually calibrate those growth parameters to get us very, very close to the real observations and the distribution of the real observations. So it's not only, of course, a single neuron that we're trying to reproduce, but we usually have many neurons of the same type. And so we want to get as close as possible to the actual distribution of those observations. And so people who are interested in this line of research are very much encouraged to have a look into these papers, and we have several more, but I just wanted to highlight here 2 recent papers in this direction. And also, we recently published a book on anti-fragile systems. So what are anti-fragile systems? Anti-fragile systems can be understood as systems that are the opposite of fragile. And so it's not really, the opposite of fragile is not really robust, because anti-fragility means that the system even gains from stochasticity and from noise. So it's a good thing for that system. And the brain can be understood as an anti-fragile system, because there is a lot of noise, of course, and stochasticity during brain development. And these are also necessary in order to create the brain architecture that we have at the end. And so we published a paper very recently with some collaborators, where we delve into also neural anti-fragile systems, such as, for example, neural connectivity. And so this is a different perspective, but it is definitely relevant. So if you're interested in this kind of more mathematical approaches, then you're very encouraged to have a look into this book. Right, so I just wanted to highlight again that the biodynamic collaboration is a multi organizational, international organization. At the moment, we are eight organizations. We started from 2, and we are very happy to collaborate also on other topics. We are working, for example, also on cancer and other complex systems that we can simulate with Biodynamo. So if you're interested, please get in touch. And I want to thank you for your attention. And if you have any questions, please don't hesitate to reach out. Thank you.